Everyone, in this video we will see HMP Shunt, Hexose monophosphate shunt. Hexose monophosphate shunt. This is also called as a pentose phosphate pathway. And this is alternative to glycolysis and TCA cycle. Alternative to glycolysis and TCA cycle. Here also in HMP shunt also oxidation of glucose takes place and it is more anabolic in nature. And during this HMP shunt, NADPH and pentoses are generated. And in this pathway, there is no utilization of ATP and there is no generation of ATP. But this is very important pathway as it produces certain important NADPH and pentoses, which are very much required for the synthesis of fatty acids and also steroids. As pentoses are required for the synthesis of nucleic acids and nucleotides. And coming to the site of the HMP shunt, it takes place in cytoplasm of the cell. And some of the tissues are more active in carrying out the HMP shunt. Those are liver, adipose tissue, then testes, erythrocytes, and lactating mammary glands. And if you see, these tissues are also very active in the synthesis of fatty acids and steroids. And whatever the NADPH that is generated in HMP shunt, that is going to be utilized for the biosynthesis of fatty acids and steroids. And this is the brief introduction regarding HMP shunt. Now we will see what are the reactions that takes place in HMP shunt. The reactions are mainly divided into two phases. One is oxidative phase and another one is non-oxidative phase. HMP shunt is divided into two phases. First one is oxidative phase. Then second one is non-oxidative phase. Non-oxidative phase. Now we will see what are the reactions that takes place in oxidative phase. So it starts with the glucose 6-phosphate. How glucose 6-phosphate is generated, that you know, when glucose is phosphorylated with the help of hexokinase, it forms the glucose 6-phosphate. This undergoes oxidative decarboxylation with the help of an enzyme, glucose 6-phosphate dehydrogenase enzyme. Which is NADP dependent enzyme, where NADPH is generated with the formation of 6 phosphoglucanolactone. Then next reaction is this 6-phosphoglucanolactone is converted to 6-phospho gluconate. This takes place with the help of glucanolactone hydrolase enzyme. And next reaction is, so this is a oxidative decarboxylation reaction, and then next reaction is again oxidative decarboxylation reaction. 6-phosphogluconate is converted into ribulose 5-phosphate. With the help of phosphogluconate dehydrogenase enzyme. Phosphogluconate dehydrogenase enzyme. This is also NADP dependent enzyme where NADPH are generated. So this is oxidative phase because oxidative reactions takes place. That's why this is oxidative phase of HMP Shunt. So HMP Shunt is also known as Hexose monophosphate pathway or Pentose phosphate pathway. The reactions are divided into two phases. One is oxidative phase and another one is non-oxidative phase. This takes place in cytoplasm, and we have seen just now the oxidative phase. Again we will see the glucose 6-phosphate undergoes oxidative decarboxylation with the help of glucose 6-phosphate dehydrogenase and forms 6-phosphoglucanolactone, and with the generation of NADPH. And then second reaction of oxidative phase is 6-phosphoglucanolactone is converted to 6-phosphogluconate with the help of glucanolactone hydrolase enzyme. And the next reaction is 6-phosphogluconate is converted to, or it undergoes oxidative decarboxylation with the generation of NADPH. It forms ribulose 5-phosphate. This is oxidative phase of HMP Shunt. Now we will see oxidative phase of HMP Shunt. Ribulose 5-phosphate, which is formed during oxidative phase, will continue in non-oxidative phase. Here, two enzymes can act on ribulose 5-phosphate. One is epimerase and another one is transketolase. When epimerase acts on ribulose 5-phosphate, it forms xylulose 5-phosphate. When transketolase acts on ribulose 5-phosphate, it forms ribose 5-phosphate. Then transaldolase. Transaldolase acts on xylulose 5-phosphate and also ribose 5-phosphate, and it acts on xylulose 5-phosphate and it transfers its 2-carbon moiety to ribose 5-phosphate so that it becomes 7 xylulose pseudoheptulose Now we have cellulose 5-phosphate, which is a 5-carbon compound, and then ribose 5-phosphate is also 5-carbon compound. Then trans aldolase acts on cellulose 5-phosphate and transfers its 2-carbon moiety to ribose 5-phosphate, and it is going to form 7-carbon compound, that is pseudoheptulose 7-phosphate, which is a 7-carbon compound, and then glyceraldehyde 3-phosphate. So this 7 pseudoheptulose 7-phosphate is 7-carbon compound, and glyceraldehyde 3-phosphate is 3-carbon compound. Then trans ketolase acts on pseudoheptulose 7-phosphate and transfers its 3-carbon moiety to glyceraldehyde 3-phosphate. So when it receives glyceraldehyde 3-phosphate receives 3-carbon fragment from the pseudoheptulose 7-phosphate, it becomes fructose 6-phosphate, and then the remaining 4 carbons are going to form as erythrose 4-phosphate. This fructose 6-phosphate enters glycolysis, and ultimately it is going to be oxidized in glycolysis as well as TCA cycle. Yeah. And what about this erythrose 4-phosphate? It should be further oxidized. So again, one more molecule of cellulose 5-phosphate participates, that is with the help of trans ketolase enzyme. So this trans ketolase transfers 2-carbon fragment from cellulose 5-phosphate to erythrose 4-phosphate. When it receives 2-carbon fragment from cellulose 5-phosphate, it becomes fructose 6-phosphate. Whereas the remaining 3 carbons are going to be formed as glyceraldehyde 3-phosphate. And we know the fate of this glyceraldehyde 3-phosphate and fructose 6-phosphate. They enter the glycolysis and TCA cycle, and further they are going to be completely oxidized. And this trans ketolase enzyme is dependent on TPP, thymine pyrophosphate, and also magnesium ions are required for these reactions. So this is a non-oxidative phase of HMP Shunt. Once again I will explain. So in oxidative phase we will get ribulose 5-phosphate, and upon this two enzymes can act. Epimerase, when epimerase acts on ribulose 5-phosphate, it is going to form xylulose 5-phosphate, and when trans ketolase acts on ribulose 5-phosphate, ribose 5-phosphate is formed. And then trans aldolase acts on cellulose 5-phosphate and transfers its 2-carbon moiety to ribose 5-phosphate and forms pseudoheptulose 7-phosphate, and then glyceraldehyde, the remaining 3 carbons are going to be formed as glyceraldehyde 3-phosphate. Then trans ketolase enzyme acts on pseudoheptulose 7-phosphate and transfers its 3-carbon moiety from pseudoheptulose 7-phosphate to glyceraldehyde 3-phosphate, and it forms fructose 6-phosphate and erythrose 4-phosphate. This fructose 6-phosphate enters TCA cycle and glycolysis and completely oxidized through this. Whereas trans-ketolase enzyme again acts on one more molecule of xylulose 5-phosphate and transfers its 2 carbons from xylulose 5-phosphate to erythrose 4-phosphate, so that it forms glyceraldehyde 3-phosphate and fructose 6-phosphate. So the non-oxidative phase is completely concerned with the interconversion of 3 carbons, 4 carbons, 5 carbons and 7 carbon monosaccharides. So this is about HMP Shunt. Thank you.